Okay. So this is an example of a pattern present in protein sequences. Okay. So again to the sequence, but another aspect, there are slightly more glutamates than aspartates in the alignment. In this case, in this E for glutamates and B for aspartates, and so there are that is 3 out of 4 sequences. There are in 3 sequences there is glutamate or glutamic acid, and in only 1 sequence out of 4 sequences there is aspartic amino acid in this sequence. Okay, so and then so what we can do, we can make profiles by adding some numbers to the problem. Okay, so consider this sequence again and the positions. Say for example, if we write all 20 amino acids starting from 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. So I have written 14 sequences out of over here, and then a number or a score for out of these, you see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Okay, so I have given because say for example a score of 20, I have given 15 score to E and 5 score to B, because E is 75% at this position number 1, and B is 25% at position number 1. So accordingly, the number has been given over here. This number has been given to position number 1. 15 to E and 5 to B. Okay? Now, at position number 2, you see that at this position, they have given equal number 10 to F and 10 to Y. Okay? F 10 to 10, because they are the F and Y are in equal number. 50% F and 50% Y. At position number 3, again we will give the same 10 to G and 10 to A, 50-50%, because there is 2 G's in this at this position and 2 A's at this position. 2 G's and 2 A's at this position. Okay. And then the position number 4, the score 20 will be given to H, because position number 4, the score H has 20 score because H is present in all sequences. So 100% score will be given to H. And now at this position number 5, you see that every amino acid at this position contains same score because it is at the same frequency. Position number 6, yes, that is the isoleucine 5, leucine 10, and valine 5. So this is how the score is divided at this position number 6. And position number 7, again 20 score to the tryptophan W because tryptophan is conserved. So this is how we basically add scoring at this position, scoring in pattern. So this basically makes a profile of a sequence at this position. Okay. So this is called profile in bioinformatics, profile of a sequence from a sequence alignment. So two things we have discussed until now, patterns and profiles. Okay. So profiles express the patterns inherent in multiple alignment of a set of sequences. So profile express the pattern which is present in multiple alignment of a set of sequence. Profile permit greater accuracy in alignments of distantly related sequences. So these profiles and patterns basically provide a greater accuracy of the alignment. Highly conserved residues are likely to be part of the active site. So, or the important site of the protein, you can say, highly conserved residues. Highly, we have discussed about conserved residues during the previous class. So the highly conserved residues, it is possible, more likely, that they would be part of the active site or important site or binding site or functional site of the protein. Conservation patterns facilitate identification of other homologous sequences. Okay, conservation patterns. Conservation patterns facilitate identification of other homologous sequences. Okay, conservation patterns are those which basically which pattern is conserved in a family of proteins using a multiple alignment. जी क्या कहना चाह रही हैं? Any question, please? जी, question? बताएंगे क्या सवाल था आपका? ओके, मैं कोशिश कर रहा हूँ कि आवाज आपको आ जाए. इंटरनेट का बाजू का मसला होता है, अभी तो कनेक्टेड है. सो आई स्टार्ट अगेन. Okay, so we were discussing about different properties of patterns and profiles. So the patterns help in classifying subfamilies within a set of homologs. So these are different characteristics of patterns. Residues with little conservation are subject to indels and are likely to be on the surface loops of the protein structure. Okay. जिन जगहों पे पैटर्न्स में कंजर्वेशन कम होती है, तो वहां पर जो है ज्यादातर गैप्स आ सकते हैं, इंसर्शन या डिलीशन हो सकती है, और वही सरफेस जो होते हैं वो जगह होती है ज्यादातर वो जो जहाँ पे कंजर्वेशन कम होती है वो प्रोटीन के स्ट्रक्चर में उसके सरफेस के ऊपर. Reliability of the protein structure prediction methods. Reliability of protein structure prediction methods, in particular homology modeling, increase use of information obtained from multiple alignments. So जब हम protein structure prediction methods पढ़ेंगे, we have not studied yet about protein structure prediction methods and homology modeling. So when basically their reliability is increased due to information obtained from multiple alignment and different patterns and profiles. So these are some information, some aspects of patterns and profiles in proteins, in sequences, either protein or DNA. I have mentioned protein sequence, but this type of pattern can also be present in DNA sequence. Okay. So based on the patterns and profiles present in DNA, present in the sequences, especially the protein sequences, a database is designed, developed, and this database is called as ProCite, which is a premier bioinformatics service for identifications of protein patterns and profiles in protein sequences. And for identification of patterns and profiles in protein sequence, this database is called as ProCite. This is a database of protein families and domains. It contains entries describing the domains, families, and functional sites, as well as amino acid patterns and signatures and profiles in them. Okay? So a pattern or profile in a protein basically represents a signature of a protein. Okay? So if of a type of protein. So if we find a pattern in a protein sequence, we can say that this is a signature of that protein. Like we basically do the signature. We basically do this, and from the signature, one person can be recognized. From his and or her signature, a person is recognized that ये इस आदमी का signature. And because of this signature, that is, we get our check approved in the banks. So in case of proteins, patterns are the signature of the protein. जिस प्रोटीन के अंदर कोई खास पैटर्न होगा, हम ये कहेंगे कि ये पैटर्न जो है ये इस प्रोटीन का सिग्नेचर है. ये प्रोटीन फलाने ये प्रोटीन फला that is belongs to that particular family of protein. Okay. Applications of the pattern and proteins include identifying possible functions of newly discovered proteins. Okay, so you have a protein sequence, you have sequenced a protein which is not present in the database, and you want to find out the properties of that proteins. So the profile search or ProCite can give us some clue about the possible function of that particular protein. And another application is to analysis of known proteins for previously undetermined activity. Okay, it is an application that we can analyze the proteins undetermined activity. What profile gives us? ProCite outputs contain information. So when we submit our sequence in ProCite, Pro site provides us information about biologically meaningful residues like active site residues, substrate binding residues, or cofactor binding residues, amino acids, and post-translational modification sites in the protein, disulfide bond site in the protein to help the identification or determination of the function of that protein. So you have the sequence and you cannot do anything with the sequence because sequence are just the letters. Okay, so it is important to identify the function and the structure of that protein. So, using the patterns and profile, we can identify the active site residues, substrate binding residues, and other amino acids in order to find out the function of that protein. Example is prediction of phosphorylation and glycosylation sites in protein sequences. This is a very important example of the finding the profile. Phosphorylation, as you know, that phosphorylation is an important post-translational modification event in the protein in which a phosphate group is attached on the protein. Phosphate group PO4 is attached to the protein. So this attachment of phosphate group is called as phosphorylation, and it is not random. कि phosphate group कहीं पर आकर भी attach हो जाए. No. Phosphate group is attached on the protein on three amino acids. Serine, threonine, and tyrosine. 3 amino acid phosphate group is attached. And not only these 3 amino group, but that is the threonine and serine and tyrosine, which is present in a pattern of sequences. So each phosphorylation site where phosphorylation occurs, where phosphate is attached with the protein, has a specific pattern of sequence of amino acid. Okay. So if we find a pattern of sequence, if we find a pattern of amino acid sequence in a protein, which we know from our previous previous knowledge that this site is suitable for phosphorylation. So we can say that this site is phosphorylation site in this protein sequence. Likewise, glycosylation. Glycosylation is another event, post-translational event in the protein. Anyone can tell me what is post-translational modification in protein? Yes, please. Post-translational modification. जी
प्रोटीन बनने के बाद जब वो साइटोसोल से बाहर जाना होता है तो उससे डिफरेंट ग्रुप ऐड होते हैं, ग्लाइकोसाइलेशन होती है, फॉस्फेट ग्रुप ऐड होता है, तो वो सब पोस्ट ट्रांसलेशन मॉडिफिकेशन होती है ताकि प्रोटीन जो है वो अपने एक्टिव फॉर्म में आ सके और वो साइटोसोल से बाहर जाए.
यस, पोस्ट ट्रांसलेशनल मॉडिफिकेशन क्या है कि एनी मॉडिफिकेशन इन प्रोटीन आफ्टर ट्रांसलेशन, आफ्टर द प्रोसेस ऑफ ट्रांसलेशन हो गई, प्रोटीन बन गई एट दी राइबोसोम. नाउ मॉडिफिकेशन, डिफरेंट ग्रुप्स आर अटैच्ड, different bonds are cleaved in protein. All these processes are called as post-translational modification. So glycosylation is a process in which a sugar molecule is attached on the protein covalently. Phosphorylation is a process in which phosphate group is attached on the protein. Okay, phosphate group is attached on the protein and sugar molecule. So, and this attachment of that is sugar molecule or phosphate is not random. It is this attachment is done, is carried out at a specific sequences, and these sequences are called as pattern, a specific pattern of sequences in the protein. And these patterns can be identified by using the Prof database service. This is the website of the Prof, and Prof is basically is over here in xpezi.org page. Okay, and if you this is a result page of Prof. Okay, and you see that this is a consensus pattern which is present in this particular example. This is an example. This is a pattern which is present in all in this sequence, which was submitted for analysis.